Okay, so I mean, if you remember in yesterday's talk, I basically said that these days there's sort of two slightly alternative philosophies for how to do mag reconstruction. One is where you do these very large-scale analyses. Typically, you're doing assembly on a single sample and then binning off that individual sample, but you'll use many such samples. So an example here that I was involved in, I think they took 7,000 human gut microbiomes, fecal microbiomes, but also some other body sites, generated mags on each of them, and then formed this huge collection of metagenome assembled genomes. The alternative, which is a thing perhaps been focusing on in my group, is where you do more focused analyses. You'll have a smaller number of samples, samples you probably generated yourself, although not necessarily. Ideally, they'd be longitudinal, so you'd have more than one sample from each community, and that really helps, as we talked about yesterday, for resolving mags. And then we use co-assemblies and binning, which is using that multiple coverage information. And then you tend to get a larger proportion of the community binned, and it's also better for doing, it's better for understanding community dynamics. But the truth is perhaps that overall you've got less representative diversity. If you want to go out and discover diversity, those large-scale binning approaches are probably the best. But if you sort of want to understand one community's dynamics, this more focused cross-sectional approach is probably better. So I'm going to talk about one example of each. I'll talk about the Sagata binning in the context of antimicrobial resistance, some reanalysis that we did. And then before that, though, I'll talk about a project that Seb and I have worked on, looking at the dynamics of anaerobic digesters. And it will give you an example, because I suppose there's, bioinformatics is complicated, all right? And on top of the binning, which itself is fairly sophisticated, we did a lot of additional bioinformatics in the AD project and in the AMR gut analyses, and I'll try to give you an overview of that additional types of methods that you could employ. Hopefully it'll be relevant to your projects. So the AD project actually grew out of a small grant from the BBSRC. We had enough funding to actually generate a year of data from 10 different anaerobic digesters around the UK. And it's fairly high resolution metagenomics because we've got metagenome samples every week for most of that year from most of these AD reactors. So you may not know what anaerobic digestion is. It's basically a biotechnology. In a way, it's like a souped up human gut, or closer perhaps to a rumen, because it has the same basic processes as occur in the gut, but with additional methanogenesis. And that means it can be used as a means for turning waste into biogas. So it's sort of got two advantages. You can get rid of unpleasant things, things like food waste or slurry on farms, but at the same time it does actually generate biogas, which can then be turned into energy. But from our viewpoint, not the viewpoint of engineers, sadly, it's a fascinating microbial community. Okay, so this is the pipeline that Seb put together. It's something called MetaHood, and he's actually going to hopefully get on to talking, hopefully doing this as a tutorial this afternoon, or at least showing you how to run the MetaHood pipeline. And it's one of two pipelines that we use in our group. So MetaHood is basically our large-scale binning pipeline. Strong, which I'll talk about this afternoon, is our sort of more focused binning plus strain resolution pipeline. Okay. So what Seb did is he had, well, in total we had 390 AD samples from these 13 reactors. We ran co-assemblies on each reactor separately. We didn't do a giant co-assembly of all reactors because firstly, it was probably beyond what we were capable of doing with our computers. And secondly, we know that there's different microbes in different reactors. So it sort of makes sense to do the co-assembly on a longitudinal basis. But then we can then use differential coverage across all the time points from each of those reactors inside Concoct to generate bins. Now, we actually used a slightly complicated strategy for deciding what was a mag, i.e. a good bin. So one of the issues with single copy core genes is it's hard to find a large set of genes that are universal to all prokaryotes. We've got 36 or so that are universal, but 36 core genes is not a lot. Okay. The reason it's not a lot is because if you think about it, the core genes enable you to predict levels of completion and contamination, but there's an assumption there that they are evenly distributed through the genomes. If they're not evenly distributed, there can be a lot of noise in your prediction. Does that make sense to people? It's quite a subtle concept that perhaps I don't have any slides on. I would draw it up on a whiteboard.
I'm assuming that's because the ribosomal proteins are usually all in the same operons or in clusters, aren't they?
Yeah,
they account for a large number of them.
So the point is, right, if you had 1,000 core genes, the single copy dual core genes would be really reliable. Okay, but there's noise. Okay, if they were imagine they're all in like 100 kilobase pairs, you could predict that a genome was perfectly complete just because you saw that 100 kilobase pairs. Okay, but it's not really complete. You've missed the rest of the genome. Okay, so the more genes you have and the more evenly distributed they are, the better the predictions of completion will be. Does that make sense to anybody?
Yeah, I mean, it's the same principle for MLST, isn't it? That you want single copy genes, but you want them scattered throughout the chromosome, not bunched together.
Yeah, and as many as possible.
Yeah.
Okay, so that's what we did here. So we used our sort of universal set to get a an initial set of mags, 7,000. Okay, and then we did taxonomic annotation with GTDB using on those initial set of potentially good mags. And once we've got taxonomic annotation, we can use better single copy core gene sets taken from CheckM, which is a program you're going to use this afternoon for evaluating bins, which are specifically tailored to 3 different groups of organisms. So we had a separate core gene set for bacteria, archaea, and then the candidate phyloradiation, which is a particular group of bacteria with very small genomes which have atypical core genes. Okay, so that meant that we could use more core genes and get more reliable predictions of bin quality, and on that basis we ended up with 5,000 mags across the reactors, which we then de-replicated because there was some sharing of mags across reactors to get our total set of 2,000 or so mags. Okay, does that pipeline make sense to people? Any questions on it? All right, well, you'll be running it this afternoon. Now, there are still some issues to be solved. Okay. Now, one of the most challenging issues we find with these studies is actually deciding what the abundance of a mag is in different samples. Okay, and this seems like a sort of obvious thing, but it's when you start to think about it, it's perhaps a bit more complicated than you might imagine. It's particularly complicated when you've done single sample binning. Okay, now if you do a co-assembly of all your samples together and do binning, then it's very easy to get the coverage of every mag in every sample, because you have the coverages of every contig in every sample from mapping the reads back. So the coverage of the mag is just the average coverages of the contigs that make up the mag. Okay. In this case, we've done something a little bit more complicated because we've done the co-assembly of the different reactors separately. So it's quite straightforward when we see once we've dereplicated, if we've got the same dereplicated mag in multiple reactors, the coverage profiles are just the coverage profiles of the mags that went into those. Okay. And this diagram is in the archive paper, so if you can, it is quite complicated, and Seb actually did all this, so he can also explain it to you. So in that case, it's fairly straightforward. We concatenate coverages, but there's still the possibility that a mag really was in a reactor, but it wasn't successfully binned and evaluated as a mag. Okay. So that's something you need to really bear in mind when you're building mags, is that particularly with short reads, it's an uncertain process. Okay, sometimes it will work, sometimes it won't. So we can estimate that in these studies we were getting about 50 or 60% of the mags that are present, which is actually really high. On single sample binnings, you might just get 5 or 10%. Okay. So how do we deal with this possibility that the mag was there, but it wasn't actually successfully binned and evaluated as a mag? So what we do is we use one of these mapping algorithms, EMAP. We take all the contigs which weren't put into mags in a reactor, and we map them against our database of mags. And if we get a certain level of the mag observed in a reactor in which we didn't successfully bin it, then we decide that in fact it is there, and we estimate its coverage from the average coverage of the contigs that have been assigned to that mag. So it's basically a way of identifying mags that were missed, in effect. And we did compare a number of different strategies for doing this. The simpler strategy, which most people use, is to just map reads onto your mag collection. But we found mapping the assembled contigs was more reliable. And we based that on the synthetic communities that we've constructed. So this is actually a problem that Seb has been working on for a couple of years, in fact. So if you want sort of more details on it, you can talk to him. He's sort of, it's one of the things it's one of his areas of expertise, I guess. Okay. All right. So we've now got these. Are there any questions on that before I move on?
But does everyone understand what you're saying there? I mean, it is quite a complicated idea, and so the idea is that if you take a sample and you try and assemble mags, metagenome assembled genomes, that process works only some of the time. Sometimes, even though the genome is there, the cells are there, when they're all smashed up and put in with everything else, you can't fish them out into a mag. But nonetheless, those sequences are present in that sample. They just haven't assembled into a mag on that day with that sample with your pipeline. But if you want to find that they're there, what you can do is you say, well, I already know this mag exists in another sample. If I map against that mag that's been created from another sample, or against a genome that you already know about from somewhere else, you can then show that that cell, that population is there, those reads are there, and it gives you more coverage. Is that explaining it right, Chris?
Yeah, pretty much. Yeah. Okay. All right. So we've got these 2,000 mags. One thing that is a standard thing to do now is to assign them using the GTDB, and then we built a de novo tree, like I briefly explained yesterday, by concatenating core genes. We can get the taxonomic assignments. We've got one of the nice things about AD reactors is you get such a diversity of organisms. So yeah, we've got a large range of phyla and a lot of novel groups, which we're showing from these reg values over here. I think we had something like 22 novel classes, in fact. So there's quite a degree of taxonomic novelty in these reactors still. But not so much, we don't focus so much on the diversity. To us, it's more about understanding the dynamics of the communities. And as I explained yesterday, you can use, once you've done this sort of high resolution MAG construction, you can then use those MAG profiles to actually understand the community dynamics. And that's what I'm showing here with NMDS plots. So as I explained yesterday, an NMDS plot is a way of plotting a higher dimensional representation down into 2 dimensions, okay? So the dimensionality of these vectors is actually 2,000, the number of mags we have, because each one is a community profile, okay? And this enables us to get a graphical summary of how the communities are changing. One thing that's quite obvious is that for most reactors, there is a clear shift over time. So AD 2.1 in particular, it starts down here and ends up over there. And that's true in most of the reactors. Here I've used a statistic called multivariate ANOVA to determine how much of the community structure actually depends on time. And in general, it's between 30 to 50%, which is actually a really large amount of community structure explained. And Friday, we'll go through all these statistics and give you examples of how to use them. So that's quite surprising and quite interesting. So we went to look into that in more detail. Now, to do that, we need to understand something about how the AD community actually functions. So as I said, it's a lot like a gut. The input is complex organic waste, and the first step in the community is hydrolysis of that. So the conversion of those polysaccharides into monomers and oligomers. And then you get fermenters, just like you would in the gut, generating short chain volatile acids, short chain fatty acids, etc. But what's different in an anaerobic digester, that would be much more like the rumens that Mark might talk about, is you get this final step. So actually methanogenesis does happen in the human gut, I think only in about 5% of people. So there's only methanogens in a small fraction of the population, whereas they're a really important part of the AD community. Basically because they're responsible for the terminal process, which is that generation of methanogen. And methanogenesis is fun because it involves something called a syntrophy, okay? So this actual process of converting, acetogenesis, is very energetically unfavourable, okay? So this is where H2 and CO2 gets turned into acetate. So you need the methanogen there to quickly convert that into methane to make the whole thing actually thermodynamically viable. So there's a really close relationship between these acetoclastic, between the acetogens and the acetoclastic methanogens. Similarly, the other methanogenesis pathway, which is autotrophic, that also depends on syntrophs, which are producing the hydrogen, or things like formate, which are then quickly converted into methane by autotrophic methanogens. So in both cases, you have a close association between a bacteria and an archaea. All methanogens are archaea, by the way. I should have said that. So that's an interesting, it's perhaps an interesting aspect of the AD community. So when you've got these mags, question is, what are they actually doing in the community? And I hope to give a talk on this on Thursday if there's time. So in order to get an idea of what each of these mags might be doing, we used a machine learning method to predict from the genome the phenotypic traits of the organisms. As I say, I'll go through this in detail on Thursday, but it's actually quite simple. You sort of treat the genome as a bag of genes. You take a set of genomes with known traits. You build a machine learning model off of that, and that enables you to infer traits for your mags. And that's what I'm showing here for just the archaea, actually, in our reactors. So we've got quite a diversity of archaea, perhaps about 40 different mags, but that's only actually a small fraction of the 2,000 mags in our community. So despite being a numerically and physiologically important part of the community, they're actually, in terms of diversity, rather low, which is another sort of interesting thing about methanogens. So the methanogens were split into 3 or 4 major groups. We've got the methanomicrobia here, and these columns show the predictions from the machine learning trait prediction. So we've used it to predict which substrate they can use for methanogenesis. So all of the methanomicrobia are coming up as autotrophic methanogens that can either use formate or hydrogen. Here we have another group, which is the methanosarcina. So that includes some from the actual class methanosarcina, which are generalist methanogens, but also some dedicated acetoclastic methanogens. And then we've got some more exotic groups. I think these are the thermoplasmata, aren't they? Which are also coming up as autotrophic. But some of these look like they use more exotic compounds as well. And this little checkerboard pattern is actually showing the abundance of the methanogens in the different reactors, averaged over the time series. So actually immediately you can see that probably the methanosarcina are the most important. They sort of split into 2 groups, either with this methanosarcina mag or a methanosarcinia mag in general. And then actually the methanomicrobia are quite specific to the different reactors, which might explain why there's so much diversity here, but we never really got around to understanding that. But anyway, I just wanted to show you how the trait prediction can give you a little bit more understanding of what the mags are doing. And as I said, we'll talk about that on Thursday. And also that gives you another way to represent the community. So this is for 2 reactors, AD 2.1 and 2.2. I'm showing those NMDS plots, showing the shift in community structure over time. But now we can sort of take a trait view of the community. So this is showing the aggregate trait abundances summed over the different mags. So you can see that there's actually, as the community shifts, there's a shift in the type of methanogenesis, which is most important. So in AD7, acetoclastic methanogenesis becomes more important over time. Whereas in AD 2.1, I suppose all types of methanogenesis are going up, but in particular the autotrophic. So yeah, it gives you another way to sort of summarise the community that perhaps has a closer connection to function. The other thing we did with these time series was try to identify syntrophies. Okay, so our hypothesis here is that if 2 organisms are syntrophs, then they'll be really strongly correlated in abundance. Okay, and so basically we took every pair of methanogens plus bacteria, and we searched for combinations that had a correlation greater than 0.9 in the time series. And that's a very high level of correlation. So here's some examples of what we observed. So I think, I'm trying to remember which way round. Yes, the red there is a bacteria, and the blue is an archaea. So you can see that the fluctuations in these 2 organisms closely follow each other. And the bacteria in this case we can assign to a group Syntrophomonas wolfei. So that's a known syntroph. The methanogen is a methanosarcinia species. So it's highly likely that these 2 are a syntrophic pair. This is quite fun because this is one of those CPR, the Patisi bacteria. It is very strongly correlating with a Methanophilia species. So this may not be syntrophy, this may be some other sort of association. People probably think the CPR are sort of like parasites on other microbes. But yeah, it's showing some form of strong association. So to try and determine whether these predicted syntrophies are real or not, again, we built another machine, we used another machine learning method to see whether we could predict whether something was a syntroph from its genome. And in fact, we could do that quite reliably with about 85% accuracy. So we could predict just straight from the genes in an organism whether it was likely to associate with a methanogen, and then we could see which genes were important in that prediction. And we get things there that are quite plausibly associated with a syntrophic lifestyle, things like oxidoreductases, but also a lot of things to do with sugar metabolism, which sort of makes sense. They might be uptaking sugars and then converting them into things like acetate or formate for the methanogenesis. Okay, so I just wanted to give you a sort of overview of one of these high resolution studies. Was there any questions on that before I go on to AMR? Okay, you're hopefully some, maybe things will become clearer when you do some of these analyses yourselves. Okay, so now I'm going to sort of go to the other end of the spectrum, which is this large-scale human gut binning that was generated in Sagata's group. So here they got over 150,000 genomes. I think about 80,000 of median was of medium quality, and they did lots of interesting analyses in the original paper. And I would recommend that you read that. But what we did was a sort of reanalysis. So one thing they didn't cover in the original paper was antibiotic resistance. Now, obviously, antibiotic resistance is a super important in the context of human health. And the question we wanted to look at was the extent to which the gut microbiome is a reservoir of antimicrobial resistance genes. And there's quite a lot of short-term studies on ARG in the gut. And there have been some larger scale studies like the one we did, perhaps using a couple of thousand samples. But there's never been a study that uses this many samples, i.e. 6,000, and also places the results in the context of the MAGs. Okay? So because we have these big MAG collections now, we can ask which species actually have the ARGs. And also, as I'll talk about, we can determine the extent to which the antimicrobial resistance genes are mobilized in the community. The other nice thing about this set of samples is that we have them from a variety of countries with different antibiotic usage regimes. So we can see how the levels of resistance associate with the antibiotic consumption in the different countries. So the bioinformatics here was actually done by somebody called Hyun Lee, who's in South Korea, who I've been collaborating with for the last couple of years. And yeah, I mean, if you thought metahood was complicated, then this is a super complex bioinformatics pipeline. But it's the sort of thing that is necessary for these large-scale analyses. So we have binning, as we've been talking about. This was actually all done by Sagata's group. So they did single sample assembly, either with metaspades or megahit. They mapped reads back, and then they did single sample binning with metabat to generate their mags, which were then evaluated and combined into this collection. We also took all the ref-seq genomes at that point. That was some 150,000. And for both the ref-seq genomes and the mags, we did gene prediction with something called Prodigal to generate ORFs. And that will be in a number of the pipelines you look at this afternoon. Once we've got ORFs, we could predict ARGs using the CARD database, which is a standard database of antimicrobial resistance genes. And we did that for both the MAGs and the reference genomes. So now got this huge collection of antimicrobial resistance genes. to make sense of that, we actually clustered those genes at different levels of sequence identity. So we generated 100% identity clusters, and this is with a program called MMSeq2, which if you ever need to do a large-scale clustering, is a really convenient piece of software. So we did clustering at 4 different levels of identity, and then we performed actually LCA on the ARGs in the same cluster. So because each ARG can be linked back to a reference genome or a MAG with a taxonomy, we can basically say in an ARG cluster, and we did this at 99%, do we see more than one species? Okay, and if we see more than one species, then we can quite reasonably say that's a mobilized gene. So if you have a set of genes which at 99% identity appear in more than one species, which should be around 95% identity, then you can say that that gene has been horizontally transmitted very recently. So it's a nice way to detect mobility in the ARGs. But we also back that up by sequence-based methods for determining whether a gene looks like it's mobile. So we again use machine learning to decide whether a contig was on a plasmid. And then we use homology searches to look for other mobile genetic elements. So we look for integrons, insertion sequences, and conjugative elements. So we've now got this whole mass of information that we can try associating with the metadata. So this is typically how you do these studies, right? So the most obvious piece of metadata in this case was the different countries. And one thing that was very striking is that there was a large variation in resistance levels in the gut across countries. So overall, the highest total abundance of resistance genes was found in Spanish samples, and the least in the Netherlands. And in terms of diversity, it was sort of similar, but here, in terms of diversity, i.e. number of ARGs that we saw in each sample, that was highest for China and France, and lowest for the USA. So the question is, what might be driving these differences? So here we went and took some statistics on antibiotic consumption that have been compiled by the World Health Organization and another organization called CDEP. So we had 2 separate sets of statistics. There's sort of a pretty good agreement between them, but they cover different sets of countries. Okay. Now, what was very striking is when we looked at gross antibiotic consumption in a country and the total abundance of ARGs, we saw a very strong correlation, with a notable exception of China. Okay, so in these associations, China was always an outlier. It always had far more resistance, or far more resistance diversity than we would expect for the level of consumption. There's possible reasons for that. It's possible that in China they're underreporting the level of resistance, or it might be that they're using a lot of antibiotics in agriculture. We also broke it down by the individual antibiotic classes, but actually only for beta-lactams, which probably are the most clinically important antibiotic class, did we see an association between the resistance in that particular class and consumption of that particular antibiotic. All right, so we also, as I said, looked at the mobilized genes. And as I mentioned when I was going through the diagram, we do that by looking at 99% clusters and looking for genes in 99% clusters that are in more than one species, which we get from the MAG or RefSeq assignments. And overall, we could assign 10% of ARG clusters as mobile, but even though they're only 10% of the clusters, those mobile genes formed the majority of each individual's resistome. So they're clearly more abundant on average than the other genes. And as I mentioned, we had Other methods for detecting whether something was on a plasmid or associated with a mobile genetic element, insertion sequence, conjugative elements, integrons, etc. So what was reassuring, these two methods are completely independent, but we saw a good agreement between the assignment of things as plasmids and whether we predicted them to be mobile from the taxonomy. So this is what we're showing here, in fact. So for the different ARG families, I'm showing the frequency that were found in multi-species clusters and the frequency which were assigned to plasmid contigs. So we have a lot of families which have a high level of plasmid association and mobility, which makes sense. We also have some families which have a lot of multi-species clusters that aren't on plasmids, which also makes sense because there are other ways to be mobile. And then a lot of things that aren't neither on plasmids nor mobile. But what we don't have a lot of are things that are on plasmids and not multi-species. So it basically means if you're on a plasmid, you're mobilized. And that was also true of those other mobile genetic elements. So here I'm showing as a distance along the genome from, say, a conjugative system, the proportion of ORFs that were identified as mobile. That's actually here. So the closer you are to one of these mobilizing elements, the more likely we are to define it as mobile based on the taxonomy. So yeah, so that was reassuring. And in terms of our comparison with the country level data, it means that we could split the ARGs up into mobilized, multi-species, single species, or plasmid associated. And yeah, what was nice then was that we can see that most of that correlation with the country level consumption is being driven by those multi-species clusters. And that's also true of the association with diversity. What's quite surprising, and we don't perhaps entirely understand, is when we look at diversity, we do see an association with single species clusters. But not when we look at the total abundance of single species clusters. And I have a theory to do for that around the ecology of the community, but yeah, not even my co-authors believe that, so I perhaps won't go into it now. How are we doing for time? If you don't want me to go on too long, is it?
Well, you've gone on for 40 minutes, just over.
Okay, fine, all right. I've got another 5 minutes then. All right, so I will talk about this other topic. So yeah, and I think it gives you, it's also quite useful because we'll see some of these ideas again on Friday, actually. So that was all about the diversity and the total abundance of the ARGs. Another way we can characterize a gut sample is with the profile of the different types of resistance. So we've got these ARG families from the CARD database, and so we can represent each sample as the abundance of ARGs in each of those families. Okay, and then again, I've used the NMDS plots to show every sample across our whole 6,000 data points. And what's very striking is that there are two distinct clusters that are formed. What we call the FAMP cluster in red here and the background cluster in blue. We call this the FAMP cluster, firstly because it's basically associated with these antibiotic resistance classes. It's associated with resistance to fluoroquinolones, aminoglycosides, peptides, and multi-drug resistance. And overall, it seems to have more resistance than you find in the background resistor type, which in healthy individuals forms the majority of samples. So this is a bit like, and we'll talk about this on Friday, the concept of enterotypes in the taxonomic structure of the human gut. It seems that there's two distinct types in terms of antimicrobial resistance profile. And finally, the last thing we did with this data set was, and because like all large-scale studies should conclude with a network, we formed what in this case is a bipartite network of bags and genes. Okay? So it's bipartite because we've got two types of things, the genes and the mags, and then we've connected them together. And what's noticeable is we've got this large central module which contains most of the commensal organisms, and then around that a variety of mostly pathogenic organisms that are less closely connected in terms of ARG sharing to the rest of the community. But those patterns that we saw in the data, we're able to assign it to this network. So the association, the FAMP resistor type, this cluster here, seems to be mostly driven by these peripheral pathogens, whilst the response to antibiotic consumption is associated with the commensal organisms, sharing those multi-species genes in this central component. All right. Well, I hope those two examples give you some ideas for what you can do with MAGs, and also perhaps some ideas in terms of the additional bioinformatics that might be relevant to your projects. And I suppose, to me, the most interesting thing about MAGs is actually understanding the community dynamics, which is a bit different, as I said, to these large-scale analyses where you focus on diversity. All right. Okay. Well, I'll finish there. Were there any questions on any of that? Is all perfectly clear, or a bit overwhelming?
Any questions? They want the slides, please.
Okay. Yeah, I'll share the links. Later. The first study is available on the archive, and we'll be putting the second one up this week. Actually, we just got it accepted for publication.
So on the second one, I'm trying to. The take-home message is that the commensals get driven to become resistant by the consumption of antibiotics and the pathogens, so that, but they do seem to be separate kind of groups. So if your gut commensals become resistant to antibiotics, that doesn't really matter to you, whereas if potential pathogens do, we're stacking up problems for the future. Is that your reading of how it is?
I think there are different timescales here, though. So I sort of agree on a short term. Perhaps it doesn't matter. It seems that if your commensals are responding to the antibiotic consumption in the population, but I think in the future it seems quite likely to me that some of.
Sorry, we lost you there. We lost the connection.
I think that's better covered.
We lost the connection there. Can you just say that again?
Well, I think in the short term you're right. Okay, in the short term the thing to worry about is whether you're in that minor FAMP resistor type or not.
Yeah. Okay.
But longer term it may well be important that the commensals are responding in this way. If you've got all this resistance in the population.
Yeah, but you haven't shown that they exchange, do they?
Spread into pathogens.
But the exchange between those commensals and the pathogens was quite minor, wasn't it?
It's definitely slower.
Yeah.
There are still connections. I agree on the short term. I think that's true. Yeah. It maybe means it's certainly less worrying than if all this resistance was seen in the pathogens, not the commensals.
Yeah, but the resistance in the pathogens is enough to worry about. And I mean, there was a paper came out the other week which tabulated the number of deaths caused by different pathogens, and E. coli was up there in the top half dozen. And for E. coli and Klebsiella to continually be acquiring resistances in the gut is a very worrying prospect. So, yeah.
Yeah, but if you think about it, we're increasingly forced to use antibiotics of last resort, isn't it?
Yeah.
So if any of that resistance appeared in these commensal organisms where there's far more diversity, and then spread into the pathogens at some point in the future, that would really be terminal, right?
Yeah. The thing that's a bit surprising, I don't see any Enterococcus on there.
It probably is there, but we may not have labelled it. It was certainly in the data.
It might be that green thing there, I don't know. But yeah, I mean, that's the other worrying one. E. coli, Klebsiella, Enterococcus. Good. There's a question. Clara's got a question.
Yes, I would like to ask, was that made on a short read sequencing or long read sequencing? And I think we are going to probably talk about that later, but how would you see the difference in resistance profile regarding those two sequencing methods?
Did you hear that, Chris?
All right. Yes, I got that. So yeah, you're right. We are going to cover that on Thursday, actually. So everything here was short read data. These large-scale collections are almost entirely short reads. On Thursday we'll talk about long reads, and we do have some data sets from mice for long reads, but not a lot on human gut yet. In terms of predicting antimicrobial resistance from the long read data, it's just going to be way easier, right? We'll have nicer contigs, we'll be able to do better annotation. And also, I suppose, when we're thinking about this bit here in particular, where we're looking at the genomic associations, this will work better with the long reads because we'll have larger contigs, right? Although the single sample assemblies weren't too bad in general. We can see we've got up to 100 kilobase pairs, right? But yeah, I mean, it's like everything. Everything works better with long reads. So ARD identification will too, I guess.
Thank you.
And yeah, I mean, looking at biosynthetic gene clusters, which you're doing, and we didn't do here, would be super interesting in this context with long reads. Right.
Okay. All right. Shall we let you go then?
I can. So I'll come back at 2:00 and see how you're getting on.
Yeah. Okay.
If you need more time with the tutorials, we can delay the strain resolution lecture, but hopefully Seb will be able to go through a bit of the snake make and automatic binning this morning. Right?
I mean, my feeling is we're in a better place today because we've got the door open and there's air circulation in the room. I mean, we've had a bit of a perfect storm, and Seb didn't have good internet access. That room we were in yesterday is so stuffy and horrible to stay in all day. So even the brightest of minds would have sort of been running through treacle in that room. I hope today are we all feeling a bit better, a bit more positive? No? Alice is not convinced, but the rest of them are smiling. We'll see. All right then, we'll see you later. Okay. Bye.
All right, bye then. Bye-bye.
Sorry, people just keep saying snake bake, and I keep hearing steak bake. I really would like a steak bake.